In this video, we will discuss about how this CRISPR screen can identify genes important for adhesion and migration. In order to do so, what it is, they compare the genes that are migrated in the transfer assay between the Chemotaxis and Chemokinesis. Basically, in one conditions, they keep the amount of FPS same in the top and bottom well of the transfer system, and in one conditions, they keep the amount of FPS high in the bottom wells, that is around 10%, and they coated this top well with fibronectin and then check the migrations of this knockout libraries, which of the genes are able to migrate, and they are able to find out that all this will affected by this IT gem. So what it is, they use the IT gem knockout, that is ITGB2 knockout, and then check its effect, the stable cell lines of the ITGB2 knockout, and check its effect in these two different migration conditions. So the control cells are adhered to the surface like this, whereas the ITVG2 knockout cells are loosely adhered. It can be seen here. Thereafter, they also checked it for the knockout cells or the guide RNAs knockouts that plays a role towards differentiation. That we have discussed in our previous video. This FLCN and mTOR1, and they can see that those cells also behave differently, or are behave like a migratory, or behave like they are in a migration or mesochemical state compared to the control. Thereafter, they check the average speed of these knockout cells compared to the control, and they are able to see that the ITVG2 knockout cells has low average speed, and also the compared to the control, and also for this FLCN knockout and mTOR1 knockout has the lower average speed compared to the control. Thereafter, they check the persistence, that is they migrate in the specific directions, and they can see that the persistence are also low in the cases where ITVG2 knockout or FLCN knockout and mTOR1 knockout. So this indicates that this guide RNAs, that is ITVG2, FLCN and mTOR1 plays a role towards migration, and the absence of these genes will cause the suppressions in the migrations. And thereafter, in the knockout libraries, they checked the relationship between chemokinesis and chemotaxis, that is the genes that are transferred through these two different conditions, and they are able to see specific correlations where R-square value is around 0.99, that can be seen here, and the highest migratory cells have this GI2 knockout, and also this PPM1F knockout. So thereafter, they check the chemotaxis, and again check the chemotaxis and this relationship, chemotaxis relationship with this serum gradient relationship or chemokinesis relationship for some of the individual knockouts that they have made based on the results that they seen from this chemokinesis and chemotaxis relationship, and they are able to see that the highest relationships are mainly they have seen it for this ATIC, and this is for the control, and also this GIT2 also migrates in the chemotaxis more compared to the serum gradient or the chemokinesis. These results are also in line with the observations that they seen from the knockout libraries, chemokinesis and chemotaxis studies. Thereafter, they check the average speed, and they can see that average speed in the case where GIT2 knockout is there, they see the high cells, the cells are migrating at a higher speed, indicating that the GIT2 has some negative effect on the or suppressing the migrations properties of the cells. So this GIT2 knockout plays an important role toward adhesions and migrations. That has been seen here, and here also we can see that in the chemotaxis and chemokinesis studies of stable cell line, we can see that GAT2 has more fraction of cells in the chemotaxis compared to the chemokinesis GAT2 knockdown cells. So in altogether, these CRISPR skins are able to identify the genes that are important for the migrations, and their knockdown will cause accelerated migrations, or their knockdown will suppress the migrations. So suppressions could be because of ITBG2, FLCN, mTOR1 knockout, and overexpressions could be because of the GAT2 knockdown, PPM1F knockdown. So this is how they come up to this point using the transfer assay. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.